He's a cell biologist and virologist who specializes in the use of advanced microscopy to study the interplay between individual viruses and cells. So to better understand the way infection works, the way the cell responds, the way the virus responds to the cell's defenses. He has a bachelor's in biology from the Catholic University of Puerto Rico, a master's in plant cell biology from NC State, and then he did his doctoral training at NC State, studying the way cardiac cells protect themselves against viral infections, and then, like I said, how viruses counteract those responses. And then he was a postdoc at UNC Chapel Hill studying the hepatitis A virus. And interestingly, he also did work at UC Davis developing genetic tools to identify host genes that are important for viral replication. So another way of interfering with the activity of a virus. So that's his background. And interestingly, he's moving away from research and into science communication. But our presentation today is about his research. So let me introduce to you Efrain Rivera-Serrano. Take it away.
Thank you very much for the kind introduction. I'm going to start sharing my screen now. So yeah, thank you for the introduction. Hopefully my goal here today is to show you all the beauty of cell-host virus interactions and how we can visualize viral infections at the cellular level. And so one of the troubles for being a virologist is that you have to update your introduction slides every few months. And so I don't have to give a lot of introduction about why viruses are important or why should we be concerned from a public health perspective, given that we're currently in a pandemic caused by a virus. But I wanted to remind you all that this is not the first time that the human species have been faced by viral outbreaks. Here are just a few recent outbreaks in the last 20 years or so caused by viruses with an RNA genome. And so here you can see the SARS-CoV-2, which is the one that hopefully we'll know that is going on right now. There's some other coronavirus here in blue. There are other viruses from different families. The ones here in the orange colors are plague viruses. So a lot of these ones are going to be arthropod as a vector. You have also enteric viruses. And so all of these have been circulating and affecting human health for many years. And these are all, of course, a concern, a global concern. If you just think about the Caribbean as a sort of isolated islands, many of them have reached the Caribbean, including dengue, chikungunya, Zika, those three being transmitted by mosquito bites, and even the current coronavirus too. And I care about the Caribbean because that's where I'm from, born and raised in Puerto Rico. And many of you, if you picture Puerto Rico, this is what you will come to mind. And it's the beach, it's an island life, very vivid. But I am actually from the other side of the island, from Ponce in the south, and the rural part of Ponce. And you might just see a lot of green here, but that is actually where I grew up. That's my house right there that still stands. And so these will come in handy here in just a minute. So the human body is constantly exposed to microbes. Every time we breathe, every time we eat, intercourse, delivery of baby, we get bitten by mosquitoes. Every single time in our daily lives, we've been exposed to microbes. Ranging from viruses to bacteria, to fungi, to parasites, and everything in between that you can imagine. So even me, as I'm speaking right now, when I'm breathing, I'm being exposed. So, but why are we constantly getting sick? We tend to focus on the times when we get sick, but we don't highlight every other day that we are not. And so why is this? And it is because our body is equipped with most of the tools necessary to fight pathogens and to fight microbes. Here's a classic video from 1950s by David Rogers at Vanderbilt. And what you have here is a blood smear. So these round things here are the red blood cells. And this big guy here is a white blood cell, a leukocyte that is chasing and going after a bacterium. And so these cells, the role is to fight bacterial pathogens, and they engulf them, eat them, and that's how we clear a lot of our microbes. Now, bacteria are really small, but our bodies are equipped with, again, many tools. In this case, we have a larger pathogen. It is a worm, parasitic worm. And what you have here is just a lot of tiny white blood cells that we have in our bodies going and attacking this big parasite. But what about an intracellular pathogen, such as viruses? Viruses live or need to go into our cells in order to replicate. The question that you have to ask yourself is, how do cells respond to an attack from within? We're not talking about white blood cells going and chasing something and attacking it. We're talking about it's within the actual cell. And so for that, we're going to take a quick step back to history, because the key here is that they are very, very small. In fact, how viruses were first known to be was by their size, by the capacity of this filtrate that was causing disease in plants. So if you were to take this sap from that was causing disease in plants and running it through a filter that would normally filter out bacteria in the flow through, whatever it was, small enough to go to the porcelain filter, you can put it into a new plant and that was still infectious. So it was smaller than a bacteria. Much later was when the term virus came to be, that derives from the Latin word meaning slimy liquid or poison. And so this is where we're at. So we have, this is a scale of size. So here our cells, our animal cells are right here. Going to the right here is decreasing in size. And you can see that the bacteria are smaller and viruses are smaller than bacteria. And that's why they were able to cross those filters. And so these are the classic images that we see when we're looking at virus structure from for identification purposes in samples. So here you have a tissue sample from a human revealing the HIV virus. Viruses are right here, that are here in red. Now, this is not very sort of appealing. So most of what we see on the news is going to be a cartoon version of this. And again, it's because they're very, very small. But you can reconstruct what you're seeing here in these black and gray images. And here's sort of the cartoon for the HIV virus, the protein shell here being the capsid and shielding inside the genetic material of the virus. And so this is important for identification of viruses, classification. Here's sort of the print screen of one of my tweets when the coronavirus structure was first revealed back in January. And I've always liked this quote, that is, a virus is a piece of bad news wrapped up in protein, protein being that shell of the virus, and bad news being the genome and what's going to come after the infection. So again, microscopy, because of the size, is a key tool in biology, and it reveals the complex universe of the cell, which is the unit of life, and it happens to be the unit that the virus needs too. So this is just a picture that I took just with a regular microscope a couple of years ago, and you might not see a whole lot here, but this is a culture of human embryonic kidney cells. And if you cannot visualize it, just imagine this, a whole lot of cells. Each cell, if you can imagine it as an egg, the cytoplasm of the cell being the white part, and the nucleus that looks more dense here being the yolk of the egg, if you will. Now, again, the problem is that viruses are obligate intracellular parasites, so we need a way to see inside the cells in order to see what the infection is doing to the host. So I was a big fan of the Magic School Bus, so we're going to go into a trip and see what goes on inside the cell during a viral infection. So this is where we're going to go back to where it all started. This is where I grew up, and it was beautiful. That's sort of why I became a scientist. But I want you all to, for 1 second, imagine how this was at night. There's no light, right? So what happens when you flip the switch and it all becomes dark, essentially in the middle of nowhere? So if you just imagine yourself in a very dark forest for 1 second, just like the slide right now, and let's say that you just lost your friends and you need to find them. And how are you going to do that? Now, there's many ways that you can try to accomplish this task, but in darkness, an easy solution will be to use light. So you might not be able to see, for example, a spider in the dark forest, but it will be easier for you to see a firefly. So same principle. Let's say that what would happen if you give to your friend a jar full of fireflies. Despite it being so dark, you will be able to see it. And the same thing if you lost many friends and each one of them is holding a jar of fireflies, even in the darkest of nights, you will be able to find them. And so that is the concept of how we can visualize infected cells here. So again, we're going to go back to those transparent cells that all we see is this black and gray images. And let's say that we want to look at the nucleus and the nucleus alone, and we want to see it in a different color. But let's say that we also want to look at other parts of the cell, such as the mitochondria and the peroxisome, another part of the cell. If let's say that we just tag each compartment a different color, so that, imagine again, if you go back to our friends, let's say that your friend A is red, your friend B is blue, Fanella is purple, and they're all roaming around, but you can tell which one it is based on the color. And so this is what it ends up looking like. And here you have a cell, for example, here in this square you have two, and the nucleus, each nucleus is in blue, and then every yellow signal that you have here is mitochondria, and in magenta, the peroxisome. So because viruses, again, live inside the cell, you can apply the same principle. And in this case, in green are viruses. And so again, I like to put cartoons for those who cannot visualize it. So here you have each one of these ones is a cell. And you can tell that the one in the middle is the only one that has very strong green signal. And that is because it is infected with a virus. Now at this resolution, all these green blobs are just a cluster of viruses that you can then pick up. But again, you can tell where the virus is, just like if it was your friend with a jar of fireflies, and the cell just being in the dark forest. And so there's a lot that you can learn using just this type of microscopy. And you get beautiful images from it. So very snapshot of what a virus needs to accomplish. It needs to find its cell, it needs to enter the cell, it needs to replicate that genetic material, it needs to make new progeny viruses to then be released and find new host. And so all of these steps you can use microscopy to visualize it, from the binding to what happens in replication, what is happening in the cell during replication, how the new viruses are being assembled and released. Now, for the next part of the talk, I want you all to also appreciate that cells are here are not bystanders. They're not just sitting there waiting for a virus to come in, replicate, and do their thing. They have alarm systems. So everything that the virus is doing, it has to do it without the actual cell noticing it and launching an antiviral response against it. So if you imagine if someone is trying to break into your house, most of us are going to have an alarm system that's going to be triggered, locks, and all those barriers to limit accessibility. So cells are no exception. They do the exact same thing. They're going to have the alarm system that are going to go off in just trying to stop it. So I will use the example of cardiac infections in the heart for two reasons: because it's my background for my PhD, and two, because it is relevant to the current pandemic. Plenty of evidence showing that COVID-19 patients have had cardiac complications, even post recovery. So it is a thing that we know that is happening. And we know that it's happening because it's not specific to just one virus. So viral myocarditis itself is defined as cardiac damage and inflammation caused by a viral infection. So you have a healthy heart, somehow the virus causes damage and inflammation in the heart, and you end up with that condition. If we just look at random autopsies, about 10% of the human population has had a previous viral myocarditis, at least post-mortem, meaning that at least one or two of you guys here in the audience listen to me, by the time that we depart this planet, we're going to have had experiences this issue. We might not know because it usually resolves in adults. Rarely it can progress to more complicated conditions, such as even to cardiac failure. But it does affect, especially the young athlete population, and that's what we're seeing in the COVID-19. And it is, in fact, the second leading cause of sudden death in young adults, the first one being car accidents. So it just happens. Again, many viruses do it, and I just updated the slide to add coronaviruses, because we know that the infections are associated with cardiac damage. Now, we really don't know how many of these viruses are doing this damage. For many viruses, it is believed that the virus itself is killing some cells in the heart, but also a lot of the pathology comes from our own immune system trying to clear the infected cell, and by doing that is destroying our own cells. So you can imagine back to the very beginning of how our white blood cells are attacking pathogens. In this case, because the pathogen is inside our cells, our white blood cells are going to have to destroy our infected cell, and that's also how we're losing cells in the heart. So back in my PhD, I use reovirus as a model because it offers a lot of powerful tools, and it's not main pathogen to us as adults. And so we can learn a lot from this virus. We have from the same virus, we have strains that are what we call myocarditic. So when you inject them into neonatal mice, they cause cardiac lesions. So you have myocarditis. And we also have strains from the exact same virus with different strain that when we do the same experiment, they don't have no effect on the heart. So it allows us to find determinants both from the virus and the host side of what is determining whether a virus is causing disease or not. We're talking about the exact same virus, just with some genetic differences. And so what we know is that the potential for this virus to cause cardiac lesions is determined by what we call the interferon response. The interferon response is something that essentially all of our cells have, and that's how they fight viruses. So it's like the alarm system. Imagine in your neighborhood, just all houses having this the same alarm system. The caveat is that it's not specific. So we're not talking about antibodies or anything. We're just talking about it's a general response. So you have the very first cell that happens to get infected with a virus. And so how that cell will respond is by inducing interferon. Now, this is a protein that gets secreted out of the cell. So imagine just you're sending the cell sending a message. Gets secreted out of the cell, and the role of interferon is to tell other cells, neighboring cells, that there's a virus present. Back to the analogy, you're setting up an alarm and you're letting your neighbors know that there's something wrong, and in hopes that they get ready for what's coming. In the cell context, they are going to start producing antivirals, so that when the new progeny viruses come out and try to infect that neighbor, they are ready to go and they can block the infection. So essentially all of our cells have the capacity to do this. And so they will block that. And so when you think about viruses, virus that cause myocarditis, they don't want this response to happen because then the infection will get blocked. So you have that correlation. A virus that induces a lot of these response is going to have a really hard time. Just imagine a virus that is in that alarm is letting the whole body know. You're never going to get past that very first rounds of infection because you will get blocked. But if we take that virus that normally would not cause any disease, so you put it in what we call wild-type mice, just regular mice, the hearts look normal. If we then take mice where this system is messed up, is deficient, we remove genes that they cannot produce this interferon, that same virus now is capable of causing disease. So that system alone is the determinant here of whether this one virus causes disease or not in the heart. And so what we care about in the heart is that the heart is uniquely vulnerable to viruses. These cells, just like most of the cells in your body, do not undergo mitosis. So the cells that you have in your heart right now are the same ones that you had as a kid. They do not divide. And so yet our heart is continuously exposed to viruses. If the virus can get into the blood, it will eventually reach the heart. And so here's sort of like a section of the heart, and in green are the myocytes, which are that muscle cell type in the heart, and they're associated with fibroblasts here in red. So you have to imagine or ask yourself, how can this organ that is essential for life rely on this interferon system that I told you every other cell type in your body has? And so for that, we use what we call primary cultures, where we were taking cells straight from the hearts of pinkies, if you will, neonatal mice, and we can just grow them in petri dish and separate muscle cells versus non-muscle cells, and just see how they behave. Now, interestingly, these cardiac muscle cells, the ones that they're almost never go into mitosis, they don't divide, they don't get replenished. They produce a lot of this interferon prior to infection. So you can imagine them as a pre-arming mechanism. Normally, I just told you that the cell has to be infected for it to produce interferon as a signal. This muscle cell types or cell type is already doing this. So it's already in that alarm pre-arming system. And so a lot of my work there was trying to determine or guess or understand how is this happening? How can these cells be producing interferon? So here's sort of like, I'm not going through all this, but this is one of the pathways that results in production of interferon. This is inside of our cell, and our cell is going to have sensors and alarms. And when all of this happens, then that produces interferon in the cell. So sort of like the big question was, is this pathway just active, just normally, in absence of infection in these unique cells? And I'll summarize all the data to just tell you that yes, it is. And so in the heart, this is the model that we had, is that in the cardiac fibroblasts, those cells that are in the heart, but they're not the muscle cell type, they divide normally, they replenish. In fact, when you have cardiac damage, they're the one that take over the lost cells. This pathway is sort of shut down, just like every other cell type in your body, waiting for a virus to produce interferon. There's no reason for it to produce interferon pre-infection. Now, cardiac myocyte, this pathway is activated. And we mapped the mechanism as to how this happens, and it all happens here at the level of the mitochondria and this protein called MAVs. So this whole pathway is active in cardiac myocytes and producing or leading to the production of interferon. And so here in this plot here on the right part of the graph, what we're showing is that fibroblasts have very little of, produce very little interferon. Myocytes, I just told you that just basal. So this is not, there's no infection here. They're producing interferon to protect themselves in case there was an infection. But if you then do the same experiments in mice that are lacking MAVs, so this protein right here, then they look just like any other cell type. So this pathway starting here from MAVs is activated in cardiac myocytes and is that's how you trigger and produce interferon. And that interferon itself is protective. And so better armed to fight a viral infection than a cardiac fibroblast, if you will. And so for now, if this was always the case and the system was working really well, and our cardiac myocytes are almost always in an alarm system, they are pre-arming themselves, meaning the virus comes, they're ready to go, they're ready to fight it. If that was the case, we would never be sick. And so this is the other side of the coin. And so again, you have cell number one being infected, producing interferon to tell the neighbor to be ready. And so if you imagine this as a cardiac myocyte, it's already producing interferon, it's already alerting its environment. But when a virus comes, it's able to block that. And reovirus is not an exception. So we have one strain that I'm gonna label here in yellow that is able to block this. And you have another strain that is not able to block it. And that correlates with their ability to cause disease in the heart. So the yellow virus right here that is able to repress the system. So even if the cell is producing interferon, that the virus produces a protein that blocks that. So that virus is able to cause myocarditis. Now the green virus that cannot, this alarm system is actually working well and it's blocking the replication, so the virus cannot cause cardiac damage. So, and this is all related to a single protein in the virus. So there's only one protein in the virus called mu2 that determines this. And I'm gonna skip through all the data, but we use microscopy to get at how is this one protein doing this and how is it shutting down interferon signaling and all that. And without microscopy, we would have never been able to do it. And so what we found is that this protein that now we're gonna label here in green, so each one of these green dots, that's where the mu2 protein is actually inside the nucleus. The nucleus here is in blue, and it's actually associated in, I'm skipping through all the data, but it's binding to a protein here in this very discrete dots inside the nucleus, that one being SRSF2. And that was with the virus that caused myocarditis. If you look at the other strain, that mu2 was not in the nucleus, and you can see that it's outside of the blue, and it was not doing anything with that protein. Inside the nucleus, you can see, so here are 4 cells. Each one of these blue blobs is the nucleus of each one of those cells, and you can see that there's 2 cells that where SRSF2, which is in red, looks squiggly, and the other one looks dotted. And that is because the middle 2 had the virus. And so this virus, what it was actually doing was sending one of its proteins to the nucleus and completely messing it up, this red protein that our cells have. And that red protein is a cell that our bodies and our cells have. And that is important for maturation of our own RNAs. From genes make RNAs, RNAs make protein. And so, but RNAs not need to mature. And so this protein is a key factor there. Yet this virus was trapping it and changing its localization, messing it up. Skipping through all the data, we also show that what the virus was doing and to the maturation of all those RNAs. But also importantly, if we take cells and we remove this factor that we found to be associated with this viral protein, what happens to infection? What happens if we just remove it? And so here are just protein levels. So we have control cells and some that we just got rid of SRSF2. And I'll summarize it, and I will tell you that the virus then replicates better, and the virus is now able to kill more cells. So we identified this protein that our cells have that somehow is controlling viral infection. The virus is trying to get it out of its regular function, but even if you deplete it, then the virus can do so much better. So this cellular factor has some antiviral functions. If you just remove it, then some of those antivirals that the protein will normally, that our cells will normally produce, the magnitude is impaired. So somehow SRSF2 is important to mount an efficient antiviral response. And this virus is trying to do its thing to get rid of it as much as it can. And the last piece is that if you just take mice that lack SRSF2, specifically in the heart, so the whole mouse has SRSF2 except for cardiac muscle cells, that is enough to cause myocarditis. So because through microscopy, we were able to identify a new protein that is involved in antiviral immunity, specifically in the heart. And so this is sort of what we have as a model. So this is our normal cell. You have this factor that I told you about here in those dots in the nucleus that is important for maturating our own RNAs in our cells. When you have a viral infection, this Mu2 protein goes into the nucleus and displaces SRSF2 and creates those squiggly lines that you saw in the microscopy. So it is affecting its function. So everything that SRSF2 was normally doing in our cells is abolished. And that results in an environment that is more suitable for viral replication. So now the virus can replicate better, and thus leading to cardiac damage. So if a different strain that cannot do this, this pathway then is fully functional and it limits its replication. So I'm going to summarize here from two sides. The first one being the host, our cells, and that being that those cardiac muscle cells are poorly replenished in our heart. Again, like those cells that you have right now are the ones that you had as a kid, and they protect themselves against pathogens by producing interferon beta pre-infection. The alarm system is ready to go, and they're ready to fight. And we identify how is this happening at the molecular level. But the system is not perfect, and viruses have found ways to counteract that. And so if we just look at reovirus as a model, those strains that are able to cause cardiac damage block the system by producing this one protein that then goes into the nucleus, messes up a host factor that is important for cardiac homeostasis to begin with, knowing that if we just remove that host in mice, that creates myocarditis. And so that'll wrap up, and I'll be taking questions after the interview. But I wanted to show that both sides of what the hosts of our cells are doing to protect themselves, but also how viruses have evolved to fight back. And so there's a lot that we don't know. We don't know about other viruses. We don't, as I said, we know about coronaviruses having association here, but we don't know much about anything. So this was sort of like a largely unexplored field about viruses in the heart that I'm sure it'll change now given the current circumstances.